We're going to talk about the hemoglobin. Hemoglobin is one of the most studied proteins in the body due to the ability to easily isolate it from red blood cells or RBCs. It comprises approximately 95% of the cytoplasmic content of RBCs. The body very efficiently carries hemoglobin in RBC, which provide protection from denaturation in the plasma and loss through the kidneys. Free non-RBC hemoglobin generated from RBC through hemolysis has a short half-life outside of the RBCs. When released into the plasma, it is rapidly salvaged to preserve its iron and amino acid components. When salvage capacity is exceeded, it is excreted by the kidneys. The concentration of hemoglobin within RBC is approximately 34 grams per deciliter, and its molecular weight is approximately 64,000 daltons. Hemoglobin's main function is to transport oxygen from the lungs to tissues and transport carbon dioxide from the tissues to the lungs for exhalation. Hemoglobin also contributes to acid balance by binding and releasing hydrogen ions and transport nitric oxide, a regulator of vascular tone. This chapter covers the structure, biosynthesis, ontogeny, regulation, and function of hemoglobin. The formation, composition, and characteristics of several these hemoglobins, namely methemoglobins, carboxyhemoglobins, and sulfhemoglobins, are also discussed in this chapter. So, we're going to discuss the hemoglobin structure. Hemoglobin is the first protein whose structure was described using X-ray crystallography. The hemoglobin molecule is a globular protein consisting of two different pairs of polypeptide chains and four hemigroups, with one hemigroup embedded in each of the four polypeptide chain. So since we talked to the heme structure, so the heme structure, heme consists of a ring of four of carbon, hydrogen, and nitrogen atom called protoporphyrins 9, with a central atom of divalent ferrous iron. Each of the four hemigroup is positioned in a pocket of the polypeptide chain near the surface of the hemoglobin molecule. The ferrous iron in each heme molecule reversibly combines with one oxygen molecule. When the ferrous iron are oxidized to the ferrous state, they no longer can bind oxygen. Oxidized hemoglobin is also called methemoglobin and is discussed later in this chapter. So we're going to discuss the globin structure. The four globin chains comprising each hemoglobin molecule consists of two identical pairs of unlike polypeptide chains, 141 to 146 amino acid each. Variation in amino acid sequences give rise to different types of polypeptide chains. Each chain is designated by a Greek letter. Each globin chain is divided into 8 helices, separated by 7 non-helical segments. The helices designated A to H contain subgroup numbering for the sequence of the amino acid in each helix and are relatively rigid and linear. Flexible non-helical segments connect the helices as reflected by their designation. NA for the sequence between the N terminus and the A helix, AB between the A and B helices, and so forth with BC, CD, DE, EF, FG, GH, and finally HC between the helix and the C terminus. Complete hemoglobin molecule. The hemoglobin molecule can be described by its primary, secondary, tertiary, and quaternary protein structure. The primary structure refers to the amino acid sequence of the polypeptide chains. The secondary structure refers to change arrangement in helices and non-helices. The tertiary structure refers to the arrangement of the helices into a pretzel-like configuration. Globin chain loop to form a cleft pocket for hemi. Each chain contains a hemi group that is suspended between the E and F helices of the polypeptide chain. The iron atom at the center of the protoporphyrins 9 ring of hemi is positioned between two histidine radicals, forming a proximal histidine bond within F8 and through the link oxygen. A close association with the distal histidine residue in E7. Globin chain amino acid in the cleft are hydrophobic, whereas amino acid on the outside are hydrophilic, which renders the molecule water-soluble. This arrangement also helps iron remains in its divalent ferrous form regardless of whether it is oxygenated or carrying an oxygen molecule or deoxygenated not carrying an oxygen molecule. The quaternary structure of hemoglobin, also called a tetramer, describes the complete hemoglobin molecule. The complete hemoglobin molecule is spherical, has 4 hemigroups attached to 4 polypeptide chain, and may carry up to 4 molecules of oxygen. The predominant adult hemoglobin, HbA, is composed of 2 alpha globin chain and 2 beta globin chain. Strong alpha 1, beta, and alpha 2 and beta 2 bond also hold the dimer in a stable form. The alpha and alpha 2 to beta bond are important for the stability of the quaternary structure in the oxygenated and deoxygenated form. A small percentage of hemoglobin A is glycated. Glycation is a post-translational modification formed by the non-enzymatic binding of various sugars to globin chain amino groups over the lifespan of the RBC. The most characterized of the glycated hemoglobins is HbA alpha, in which glucose attached to the N-terminal valine of the beta chain. Normally, about 4 to 6% of HbA circulates in the form of A1C form. In uncontrolled diabetes mellitus, the amount of A1C is increased proportionally to the mean blood glucose level over the preceding 2 to 3 months. Now we're going to talk about the hemoglobin biosynthesis. Hemibiosynthesis occurs in the mitochondria and cytoplasm of bone marrow erythrocytes precursors. Beginning with the pronormoblast through the circulating polychromatic, also known as polychromatophilic erythrocytes. As they lose their ribosomes in mitochondria, location of the citric and tricarboxylic acid cycle, mature erythrocytes can no longer make hemoglobin. Hemibiosynthesis begins in the mitochondria with the condensation of glycine and succinyl coenzyme A, catalyzed by amino-levulinic synthase to form amino-levulinic acid or ALA. In the cytoplasm, amino-levulinic acid dehydratase, also known as porphobilinogen synthase, converts ALA to porphobilinogen PBG. PBG undergoes several transformations in the cytoplasm from hydroxymethylbilane to coproporphyrinogen 3. This pathway then continues in the mitochondria until in the final step of production of heme, iron combines. So more like this. With protoporphyrins 9 in the presence of ferrosilicase or hemisynthase to make heme, transferrin, a plasma protein, carries iron in the form to developing erythroid cells. Transferrin binds to transferrin receptors on erythroid precursor cell membrane, and the receptors and transferrin with bound iron are brought into the cell in an endosome. Acidification of the endosome releases the iron from transferrin. Iron is transported out of the endosome and into the mitochondria, where it is reduced to the ferrous state and is united with protoporphyrins 9 to make heme. Heme leaves the mitochondria and is joined to the globin chains in the cytoplasm. Now we're going to discuss the globin biosynthesis. There are 6 structural gene codes for 6 globin chains. The alpha and the L-globin gene are on the short arm of chromosome 16. The E, the gamma, and distinct and beta globin gene cluster is on the short arm of chromosome 11 in the human genome. There is 1 copy of each globin gene per chromatid, for a total of 2 genes per diploid cell, with the exception of alpha and gamma. There are 2 copies of the alpha and gamma globin gene per chromatid, for a total of 4 genes per diploid cell. The production of globin chain takes place in erythroid precursor from the pronormoblast through the circulating polychromatic erythrocytes, but not in the mature erythrocytes. Transcription of the globin chain to messenger ribonucleic acid, or the mRNA, occurs in the nucleus, and translation of mRNA to the globin polypeptide chains occur on ribosomes in the cytoplasm. Although transcription of the alpha globin gene produces more mRNA than the beta globin gene, there is less efficient translation of the alpha globin mRNA. Therefore, the alpha and beta chain are produced in approximately equal amounts after translation is complete. The chain are released from the ribosomes in the cytoplasm. Now we're going to discuss about the hemoglobin assembly. After the release from ribosome, each globin chain binds to a heme molecule, then form a heterodimer. The non-alpha chains have charge differences that determines their affinity to bind to the alpha chains. The alpha chain has a positive charge and has the highest affinity for a beta chain due to its negative charge. The gamma globin chain has the next highest affinity, followed by this globin chain. I don't know the Greek letter for it. There are 2 heterodimers then combined to form a tetramer. This completes the hemoglobin molecule. 2 alpha and 2 beta chain form HbA, the major hemoglobin presence from 6 months of age through adulthood. HbA2 contains 2 alpha and 2 this chain, owing to a mutation in the promoter region of this globin chain. Production of the this chain polypeptide is very low. You can look at the papers na lang. Consequently, HbA2 comprise less than 3.5% of total hemoglobin in adults. HbF contains 2 alpha and 2 gamma chains. In healthy adults, HbF comprise 1 to 2% of total hemoglobin, and it is present only in a small portion of the RBC. Uneven distribution. These RBCs with HbF are called F or AF cells. The various amino acid that comprise the globin chain affects the net charge of the hemoglobin tetramer. Electrophoresis and high-performance liquid chromatography, or HPLC, are used for fractionation, presumptive identification, and quantification of normal hemoglobin and hemoglobin variants. Molecular genetic testing of globin gene DNA provides definitive identification of variant hemoglobins. Now let's going to talk about the hemoglobin ontogeny. Hemoglobin composition differs with prenatal gestation time and postnatal age. Hemoglobin changes reflects the sequential activation and inactivation or switching of the globin genes, progressing from the dis and to the alpha chain on chromosome 16 and from the epsilon to the gamma and this chain again and beta globin gene on chromosome 11. This gene and epsilon globin chain are normally appears only during the first 3 months of embryonic development. These two chains, when paired with the alpha and gamma chain, form the embryonic hemoglobin. During the second and third trimester of fetal life and at birth, HbF or D has 2 alpha and 2 gamma is the predominant hemoglobin. By 6 months of age and through adulthood, HbA or contains alpha 2 alphas and 2 betas is the predominant hemoglobin with small amount of HbA2 or 2 alpha and this 2 in this again and HbE F. It is presents the reference interval for the normal hemoglobin fraction at various age. Mechanism that controls the switching from gamma chain production to beta chain production or YB switching are discussed in the next section. Now we're going to discuss the regulation of hemoglobin production. Let's start with the hem regulation. The key rate limiting steps in hemosynthesis is the initial reaction of glycine and succinylene CoA to form ALA, catalyzed by ALA synthase. Heme inhibits the transcription of the ALA synthase gene, which lead to a decrease in heme production, a negative feedback mechanism. Heme inhibits other enzymes in the biosynthesis pathway, including ALA dehydrase and PBG deaminase. A negative feedback mechanism by heme or substrate inhibition by protoporphyrins 9 is believed to inhibit the ferrous chelatase enzyme. Conversely, an increased demand for heme induces an increased synthesis of ALA synthase. We're going to discuss the globin regulation. Globin synthesis is highly regulated so that there is a balanced production of globin and heme. This is critical because an excess of globin chain, protoporphyrins 9 or iron can accumulate and damage the cell, reducing its lifespan. Globin production is mainly controlled at the transcription level by a complex interaction of deoxyribonucleic acid DNA sequence, cis-acting promoter, enhancers and silencers, and soluble transcription factor like trans-acting factors that binds to DNA or to one another to promote or suppress transcription. Initiation of transcription of a particular globin gene requires the promoter DNA sequence immediately before the 5 ends of or the beginning of the gene. A key transcription factor called propyl-like factor 1 or KLF1. Then a number of other transcription factors such as GATA1, Icarus, TAL1, P45, NFE2, and LDB1. And the next step is an enhancer region of DNase 1 hypersensitive nucleic acid sequence located more than 20 kilobases upstream before the 5S start site of the gene from the globin gene called the locus controlled region or LCR. For example, to activate transcription of the beta-globin gene in the beta-globin gene cluster on chromosome 11, the LCR, the promoter for the beta-globin gene, and various transcription factors join together to form a three-dimensional active chromosomes hub or ACH, with KLF1 playing a key role in connecting the complex. Because the LCR is located a distant upstream from the beta-globin gene complex, a loop of DNA is formed when the LCR and beta-globin gene promoter join together in the chromosome hub. The other globin gene in the cluster, like epsilon, gamma, and this again, are maintained in the inactive state in the DNA loop, so only the beta-globin gene is transcribed. Crouple-like factor 1 also plays a key regulatory role in the switch from gamma chain to beta chain production, or the YB switching, that begins in late fetal life and continues through adulthood. The KLF1 is an exact match for binding to the DNA promoter sequence of the beta-globin gene, while the gamma-globin gene promoters has a slight different sequence. This results in a preferential binding to and subsequent activation of transcription of the beta-globin gene. The KLF1 also regulates the expression of repressors of gamma-globin gene transcription such as BCL11A and MYB. Globin synthesis is also regulated during translation when the mRNA coding for the globin chain associates with ribosomes to produce the polypeptide. Many protein factors are required to control the initiation, elongation, and termination steps of translation. Heme is an important regulator of globin mRNA translation and at the initiation step by promoting the activation of a translation initiation factor and inactivating its repressors. Conversely, when the heme level is low, the repressor accumulates and inactivates the initiation factor, thus blocking translation of the globin mRNA. Systemic regulation of erythropoiesis, we're going to discuss about this. When there is an insufficient quantity of hemoglobin, or if the hemoglobin molecule is defective in transporting oxygen, tissue's hypoxia occurs. The hypoxia is detected by the peritubular cells of the kidney, which responds by increasing the production of erythropoietin, or EPO. EPO increases the number of erythrocytes produced and released into the periphery. It also accelerates the rate of synthesis of erythrocytes components, including hemoglobin. Although each laboratory must establish its own reference interval based on their instrumentation, methodology, and patient population in general, reference interval for hemoglobin concentration are as follow. Men has 14 to 18 grams per deciliter, or 140 to 180 grams per liter, while for women it has 12 to 15 grams per deciliter. If in the other methods naman, it is 120 to 150 grams per liter. While for newborn, it has 16.5 to 21.5 grams per deciliter, or 165 to 215 grams per liter. The reference interval for infants and children varies according to age group. Individuals living at high altitude have slightly high levels of hemoglobin as a compensatory mechanism to provide more oxygen to the tissue in the oxygen-thin air. Tables on the inside front covers of this text provide reference interval for all age group. Now let's going to discuss about the hemoglobin function. So we going to discuss the oxygen transport. The function of hemoglobin is to readily bind oxygen molecules in the lungs, which require high oxygen affinity. to transport oxygen and to efficiently unload oxygen to the tissues which require low oxygen affinity. During oxygenation, each of the four heme iron atoms in a hemoglobin molecules can reversibly bind one oxygen molecule. Approximately 1.34 ml of oxygen is bound by each gram of hemoglobin. The affinity of hemoglobin for oxygen relates to the partial pressure of oxygen or PO2, often defined in terms of the amount of oxygen needed to saturate 50% of hemoglobin, called the P50 value. The relationship is described by the oxygen dissociation curve of hemoglobin, which plots the percent oxygen saturation of hemoglobin versus the PO2. The curve is sigmoidal, which indicates low hemoglobin affinity for oxygen at low oxygen tension and high affinity for oxygen at high oxygen tension. Cooperation among hemoglobin subunits contributes to the shape of the curve. Hemoglobin that is completely deoxygenated has little affinity for oxygen. However, with each oxygen molecule that is bound, there is a change in the conformation of the tetramer that progressively increase the oxygen affinity of the other heme subunits. Once one oxygen molecules bind, the remainder of the hemoglobin molecules quickly becomes fully oxygenated. Therefore, with the high oxygen tension in the lungs, the affinity of hemoglobin for oxygen is high, and hemoglobin becomes rapidly saturated with oxygen. Conversely, with the relatively low oxygen tension in the tissues, the affinity of hemoglobin for oxygen is low, and hemoglobin rapidly releases oxygen. Normally, pressure oxygen of approximately 27 millimeters mercury results in 50% oxygen saturation of the hemoglobin molecule. If there is a shift of the curve to the left, 50% oxygen saturation of hemoglobin occurs at a pressure oxygen of less than 27 millimeters per mercury. If there is a shift of the curve to the right, 50% oxygen saturation of hemoglobin occurs at a pressure oxygen 2 higher than 27 mm per mercury. The reference interval for arterial oxygen saturation is 96% to 100%. If the oxygen dissociation curve shifts to the left, a patient with arterial and venous pressure oxygen levels in the reference interval 80 to 100 millimeters mercury arterial and 30 to 50 millimeters mercury venous will have a higher percent oxygen saturation and a high affinity for oxygen than a patient for whom the curve is normal. With a shift in the curve to the right, a lower oxygen affinity is seen. In addition to the pressure oxygen, shifts of the curve to the left or right occurs if there are change in the pH of the blood. In the tissues, a lower pH shifts the curve to the right and reduces the affinity of hemoglobin for oxygen, and the hemoglobin more readily releases oxygen. A shift in the curve due to the change in pH or hydrogen ion concentration is termed the Bohr's effect. It facilitates the ability of hemoglobin to exchange oxygen and carbon dioxide and is discussed later. The concentration of 2 to 3 biphosphoglycerate or 2 to 3 BPG for formerly 2 to 3 diphosphoglycerate also has an effect on oxygen affinity. In the deoxygenated state, the hemoglobin tetramer assumes a tense or T conformation that is stabilized by the binding of 2 to 3 BPG between the beta-globin chains. The formation of salt bridges between the phosphates of 2 to 3 BPG and positively charged group on the globin chains further stabilize the tetramer in the T conformation. The binding of 2 to 3 BPG shifts the oxygen dissociation curve to the right, favoring the release of oxygen. In addition, a lower pH and a higher PCO in the tissue further shifts the curve to the right, favoring the release of oxygen. A hemoglobin binds oxygen molecule. A change in conformation of the hemoglobin tetramer occurs with a change in hydrophobic interaction at the alpha-beta contact point. A disruption of the salt bridges and release of 2 to 3 BPG. A 15-degree rotation of the alpha-beta dimer relative to the alpha-beta dimer occurs along the alpha-beta contact point. When the hemoglobin tetramer is fully oxygenated, it assumes a relaxed or R state. Clinical conditions that produce a shift of the oxygen dissociation curves to the left include a lower body temperature due to external causes, multiple transfusion of stored blood with depleted 2 to 3 BPG, alkalosis, and the presence of hemoglobin variants with a high affinity for oxygen. Condition producing a shift of the curve to the right includes increased body temperature, acidosis, the presence of hemoglobin variants with a low affinity for oxygen, and an increased 2 to 3 BPG concentration in response to hypoxic conditions such as high altitude, pulmonary insufficiency, congestive heart failure, and severe anemia. The sigmoidal oxygen dissociation curve generated by normal hemoglobin contrasts with myoglobin's hyperbolic curve. Myoglobins present in cardiac and skeletal muscle is a 17,000 Dalton monomeric oxygen-binding hemoprotein. It binds oxygen with greater affinity than hemoglobin. Its hyperbolic curves indicates that it releases oxygen only at very low partial pressure, which means it is not as effective as hemoglobin in releasing oxygen to the tissues at physiologic oxygen tension. Myoglobin is released into the plasma when there is a damage to the muscle in myocardial infarction, trauma, or severe muscle injury, called rhabdomyolysis. Myoglobin is normally excreted by the kidneys, but levels may become elevated in renal failure. Serum myoglobin level aids in diagnosis of myocardial infarction in patients who have no underlying trauma, rhabdomyolysis, or renal failure. Myoglobin in the urine produces a positive results on the urine dipstick test for blood. This must be differentiated from a positive results caused by hemoglobin. HbF, or the fetal hemoglobin, the primary hemoglobin in newborn, has a pressure 50 of 19 to 21 millimeters mercury, which resulted in a left shift of the oxygen dissociation curve and increased affinity for oxygen relative to that of HbA. This increased affinity for oxygen due to its weakening ability to bind 2 to 3 BPG. There is only one amino acid difference in a critical 2 to 3 BPG binding site between the beta chain and between the gamma chain and the beta chain that accounts for this difference in binding. In fetal life, the high oxygen affinity of HbF provides an advantage by allowing more effective oxygen withdrawal from the maternal circulation. At the same time, HbF has a disadvantage in that it delivers oxygen less readily to tissues. The bone marrow in the fetus and newborn compensate by producing more RBCs to ensure adequate oxygenation of the tissues. This response is mediated by erythropoietin. Consequently, the RBC count, hemoglobin concentration, and hematocrit of a newborn are higher than adult values. values are on the inside front cover, but they gradually decrease to normal physiologic level by 6 months of age as the gamma beta switching is completed, and most of the each of the fetal hemoglobin is replaced by HbA. Now we're going to discuss about the carbon dioxide transport. A single crucial function of hemoglobin is the transport of carbon dioxide in venous blood. The carbon dioxide diffuses into the red blood cell and combines with water to form carbonic acid or H2CO3. This reaction is facilitated by the RBC enzyme carbonic anhydrase. Carbonic acid then dissociates to release H and bicarbonate or HCO3. The H from the second reaction bind oxygenated hemoglobin HbO2, and the oxygen is released from the hemoglobin due to the Bohr effect. The oxygen then diffuses out of the cell into the tissue. As the concentration of negatively charged bicarbonate increases, it diffuses across RBC membrane into the plasma. Chloride, also negatively charged, diffuse from the plasma into the cell to maintain electron neutrality across the membrane. This is called the chloride shift. In the lungs, oxygen diffuses into the cell and binds to the oxygenated hemoglobin or HHB due to the high oxygen tension. H is released from hemoglobin and combines with bicarbonate to form carbonic acid. Carbonic acid is converted to water and CO2. The latter diffuses out of the cell and is expelled by the lungs. As more bicarbonate diffuses into the cell to produce carbonic acid, chloride diffuses back out into the plasma. Approximately 85% of the CO2 produced in the tissues is transported by hemoglobin as H. In this capacity, hemoglobin provides a buffering effect by binding and releasing high H. A small percentage of carbon dioxide remains in the cytoplasm, and the remainder binds to the globin chain as carboamino group. We're going to discuss the nitric oxide transport. It's a third function of hemoglobin, involves the binding, inactivation, and transport of nitric oxide. Nitric oxide is secreted by vascular endothelial cell and causes relaxation of the vascular wall, smooths muscle, and vasodilation. When released, free nitric oxide has a short half-life, but some enters the RBCs and can bind to cysteine in the beta chain of hemoglobin, forming S-nitrous hemoglobin. Some investigators purpose that hemoglobin preserves and transports nitric oxide to hypoxic microvascular areas, which stimulates vasodilation and increased blood flow. Hypoxic vasodilation. In this way, hemoglobin may work with other systems in regulating blood flow to microvascular areas by binding and inactivating nitric oxide, causing vasoconstriction and decreased blood flow. When oxygen tension is high in releasing nitric oxide, causing vasodilation and increased blood flow when oxygen tension is low. This theory is universally accepted, and the roles of hemoglobin, endothelial cell, and nitric oxide in regulating blood flow and oxygenation of the microcirculation are still being investigated. Now let's discuss the dishemoglobins. These hemoglobin are the dysfunction hemoglobins that are unable to transport oxygen, includes methemoglobins, sulfhemoglobins, and carboxyhemoglobins. These hemoglobins forms and may accumulate to toxic levels after exposure to certain drugs or environmental chemicals or gases. The offending agent modifies the structure of the hemoglobin molecules, preventing it from binding oxygen. Most cases of this hemoglobinemia are acquired. A small fraction of methemoglobinemia causes are hereditary. So now let's discuss the methemoglobin. Methemoglobin or MET-HB is formed by the reversible oxidation of heme iron to the ferric state or Fe+. Normally, a small amount of methemoglobin is continuously formed by oxidation of iron during the normal oxygenation and the oxygenation of hemoglobin. However, methemoglobin reduction systems, predominantly the NADH cytochrome B5 reductase 3 NADH methemoglobin reductase pathway, normally limits its accumulation to only 1% of total hemoglobin. Methemoglobin cannot carry oxygen because the oxidized ferric iron cannot bind it. An increase in the methemoglobin levels result in decreased delivery of oxygen to the tissues. In individuals with methemoglobin levels less than 25% are generally asymptomatic. If the methemoglobin level increased to more than 30% of total hemoglobin, cyanosis, bluish discoloration of skin and mucous membranes, and symptoms of hypoxia, dyspnea, headache, vertigo, change in mental status occurs. Level of methemoglobin greater than 50% can lead to coma and death. An increase in methemoglobin called methemoglobinemia can be acquired or hereditary. The acquired form, also called the toxic methemoglobinemia, occurs in normal individual after exposure to an exogenous oxidant such as nitrites, primaquine, dapsone, or benzocaine. As the oxidant overwhelmed the hemoglobin reduction system, the level of methemoglobin increases and the patient may exhibit cyanosis and symptoms of hypoxia. In many cases, withdrawal of the offending oxidants is sufficient for a recovery. But if the level of methemoglobin increases to 30% or more of total hemoglobin, intravenous methylene blue is administered. The methylene blue reductase the methemoglobin ferric iron to the ferrous state through the NADPH methemoglobin reduction pathway that involves glutathione reductase and glucose-6-phosphate dehydrogenase. In life-threatening cases, exchange transfusion may be required. Hereditary cause of methemoglobinemia are rare include mutation in the gene for NADPH, cytochrome B5 reductase 3, or CYB5R3, resulting in a diminished capacity to reduce methemoglobin and mutation in the alpha, beta, or gamma globin gene, resulting in a structurally abnormal polypeptide chain that favors the oxidized ferric form of iron and prevent its reduction. The methemoglobin produced by the latter group is called M-hemoglobin or HbM. The HbM inherited in an autosomal dominant pattern, with methemoglobin comprising 30 to 50% of total hemoglobin. There is no effective treatment for this form of methemoglobinemia. Cytochrome B reductase deficiency is an autosomal recessive disorder, and methemoglobin elevation occurs in individuals who are homozygous or compound heterozygous for a CYB5R3 mutation. Most individuals with HbM or homozygous cytochrome B5 reductase deficiency maintain methemoglobin levels below 50%. They have cyanosis but only mild symptoms of hypoxia that do not require treatment. Individual heterozygous for the CYB5R3 mutation have normal levels of methemoglobin but develop methemoglobinemia, cyanosis, and hypoxia when exposed to an oxidant, drug, or chemical. Methemoglobin is assayed by spectral absorption analysis instruments such as the CO oximeter, Methemoglobin shows an absorption peak at 630 nanometer. With high levels of methemoglobin, the blood takes on chocolate brown color and does not revert back to the normal red color after oxygen exposure. The methemoglobin in HBM disease has different absorption peaks depending on the variants. Hemoglobin electrophoresis, high-performance liquid chromatography, and DNA mutation testing are used for identification of hemoglobin M variants. Cytochrome B reductase 3 deficiency is diagnosed by enzyme assay and DNA mutation testing. Now let's discuss about the sulfahemoglobin. Sulfahemoglobin is formed by the irreversible oxidation of hemoglobin by drugs such as sulfonamide, pinacetin, nitrites, and phenylhydrazine, or exposure to sulfur chemical in industrial or environmental setting. It is formed by the addition of a sulfur atom to the pyrrole ring of heme and has a greenish pigment. Sulfahemoglobin is ineffective for oxygen transport in patients with elevated levels present with cyanosis. Sulfahemoglobin cannot be converted to normal HbA. It persists for the life of the cell. Treatment consists of prevention by avoidance of the offending agent. Sulfahemoglobin has a similar peak to methemoglobin on a spectral absorption instrument. The sulfahemoglobin spectral curve, however, does not shift when cyanide is added, a feature that is used to distinguish it from methemoglobin. Carboxyhemoglobin or COHB results from the combination of carbon monoxide with hemi-iron. The affinity of carbon monoxide for hemoglobin is 240 times that of oxygen. Once one molecule of carbon monoxide binds to hemoglobin, it shifts the hemoglobin-oxygen dissociation curve to the left, further increasing its affinity and severely impairing release of oxygen to the tissues. Carbon monoxide has been termed the silent killer because it is an odorless and colorless gas, and victims may quickly become hypoxic. Some carboxyhemoglobin is produced endogenously, but it normally comprises less than 2% of total hemoglobin. Exogenous carbon monoxide is derived from the exhaust of automobiles, tobacco smoke, and from industrial pollutants such as coal, gas, and charcoal burning. In smokers, COHB levels may be as high as 15%. As a result, smokers may have a higher hematocrit and polycythemia to compensate for the hypoxia. Exposure to carbon monoxide may be coincidental, accidental, or intentional, suicidal. Many deaths from house fires are the results of inhaling smokes, fumes, or carbon monoxide. Even when heating systems in the home are properly maintained, accidental poisoning with carbon monoxide may occur. Toxic effects such as headache, dizziness, and disorientation begins to appear to blood levels of 20% to 30% COHB. Levels of more than 40% of total hemoglobin may cause coma, seizure, hypotension, cardiac arrhythmias, pulmonary edema, and death. Carboxyhemoglobin may be detected by spectral absorption instrument at 540 nanometers. It gives blood a cherry red color, which is sometimes imparted to the skin of victims. A diagnosis of carbon monoxide poisoning is made if the COHB levels is greater than 3% in non-smoker and greater than 10% in smoker. Treatment involves removal of the patient from the carbon monoxide sources and administration of 100% oxygen. The use of hyperbaric oxygen therapy is controversial. It is primarily used to prevent neurologic and congenital impairment after acute carbon monoxide exposure in patients whose COHB level exceeds 25%. Now let's talk about the hemoglobin measurement. The cyan-methemoglobin method is the reference method for hemoglobin assay. A lysing agent present in the cyan-methemoglobin reagent frees hemoglobin from RBCs. Free hemoglobin combines with potassium ferric cyanide contained in the cyan-methemoglobin reagent, which converts the hemoglobin ion from the ferrous to the ferric state to form methemoglobin. Methemoglobin combines with potassium cyanide to form the stable pigment cyan-methemoglobin. The cyan-methemoglobin color intensity, which is proportional to hemoglobin concentration, is measured at 540 nanometers spectrophotometrically and compared with the standard. The cyan-methemoglobin method is performed manually but has been adapted for use in automated instrument. Many instruments now use sodium lauryl sulfate or SLS to convert hemoglobin to SLS methemoglobin. This method does not generate toxic waste. Hemoglobin electrophoresis and HPLC are used to separate the different types of hemoglobin such as HbA, and F. And I think we're all done. So yeah, so that's it. So thank you for watching this video.